Welcome. This animation is meant to prepare you for the first day of the laboratory in which we purify and amplify a non-coding portion of your 16th chromosome. The first day is the purification of your own DNA from your cheek cells. The second day we will use a device called a thermal cycler to run the polymerase chain reaction and generate many copies of the portion of DNA we're studying. If we don't get it right today, the remaining days are an exercise in futility. The animation is meant to support your understanding of the procedure you've already read. What you are looking at is a homologous pair of your 16th chromosome. We have these pairs because it took two to make you. Located on the 16th chromosome is a segment known as a LU PV92. ALU segments, like the one we are studying, are only found in primates, and ALU PV92 is specific to humans. So there's all kinds of cool stuff we can learn about our own origins from studying segments like this one. Because you may or may not have inherited this ALU segment, it creates a situation where there are two versions that result in three different genotypes floating around in the global human population. The ALU PV92 element is relatively small. It's only 300 base pairs. If you have ALU PV92, it adds 300 base pairs to the 415 that are already there, for a total of 715. If you don't, then this portion of your 16th chromosome is just 415 base pairs. So basically there's a long one that we designate with a plus sign and a shorter one that we designate with a minus sign. So your genotype could be plus plus, giving you two long ones, plus minus, making you heterozygous, or you could be homozygous for the short one, and be minus minus. So this lab is pretty cool in that you can find out your genotype, which is nice to do in class. We can use analysis of our class data to learn something about our ancestry, and in a general sense determine if evolution is occurring in a population. We'll do all this later on. Let's get started now. Alrighty. So, keep in mind that we need to come in and immediately begin. This is a lot to do in regular class period. The first step is to isolate your cheek cells. I will provide you with a Dixie cup partly filled with 10 milliliters of a dilute salt solution. You need to swish this vigorously in your mouth for at least a minute. The more vigorous you swish, the more cells will come off the inside epithelium of your cheeks. Please don't swallow the solution and don't be super dramatic. It's not that gross. Alrighty, so after swishing, you'll carefully spit the nasty mixture of whatever was in your mouth back into the cup, and I've listed a few of the ingredients, so we obviously want to be careful with our cups so they don't spill. Go ahead and obtain a microtube and a plastic pipette. Write a 4-digit pin on the side and lid of the tube with a Sharpie. Be original and mature in your number choice. If you use your birthday, make it month and day. I'm pretty sure I wasn't the only person born in December 1967. Swirl the cup and draw some of the solution into the pipette. You will transfer 1 ml of the solution to the labeled tube. Use the graduations on the tube to make sure you have the correct volume. Okay, so now we're ready to start isolating our cells. To do this, we will use a microcentrifuge. These are powerful little machines that spin the microtubes at high speeds. It is very important that when we place the tubes in the rotor that we follow some rules. We need to cap the tube tightly. When you place your tube, look at the number of the hole in the rotor you've put the tube in. It always amazes me year after year how some of your brains shut down and you manage to remove someone else's tube. Do me a favor and look at the number on the rotor and the number on your tube. The microcentrifuge needs to be balanced. Each tube must have the same volume. It's 1 milliliter at this point, and we must arrange tubes in the rotor in a certain way. These arrangements can only be achieved with 2, 3, 4, or 6 tubes. Okay, so we can go like this, or we can go like this, or we can go like this. Okay, you can't have lopsided rotors by putting all the tubes next to each other if you have 2, 3, or 4 tubes. They can't all be next to each other. You can't have 1 tube, you can't have 5 tubes. If you are a lone spinner, you can make a blank tube, and this is a tube with the same volume of water in it, and then you have at least 2 tubes to put in the rotor. And make sure long hair and jewelry are clear of the centrifuge before you start spinning. Once loaded, you'll spin for 1 minute at the highest speed. When you remove your, that's right, I said your tube, you should be able to visualize the sacred cell pellet. This cell pellet is now the center of your universe. Hold the tube up to the light and make sure that you have a pellet. If you're not sure, call me over. So we need to get rid of most of the liquid layer called the supernatant. It is important to do this very carefully so you don't lose the pellet. If you do, you need to start over. You can draw out the liquid with your pipette, or you can try to pour off the supernatant back into your cup. So the next step is to resuspend the cell pellet in the small volume of supernatant that you left behind. You can do this by dragging your tube across the microtube rack a few times. The pellet should be gone and the solution should look cloudy. Come to me and obtain your next tube and write your pin on it, lid and sides. You will be adding your cell solution to this new tube. Now go to the first micropipetting station. We've gone over rules for use in class. So you know not to get all plunger and ejector happy. You make sure you have a tip on, don't dial the micropipette out of the range, and don't hold it tip end up, and make sure to dial the P200 to 30 microliters. And we'll drop some of that cell solution. Okay? So once you've drawn up 30 microliters of your cell suspension, you're going to transfer it to the new tube that has this stuff in it called Chelex. Okay, here's a major source of error, so we need to be careful that you put your cells into the Chelex tube and not vice versa. These Chelex beads are chemical magic and will help purify your DNA in the steps that follow. Now you're ready to transfer your tube to the boiling water bath. Close your lid tightly and secure it with a cap lock. You can also poke a hole using a push pin to vent the tube if you want. The point is that we want to make sure that the pressure building up in the tube doesn't cause it to burst open. Make sure you can clearly read the number you've written on the lid. Sometimes the number on the side can come off in the water bath. We have these cool floating racks. You're going to place your numbered, locked, and vented tube into the rack. When it's full, I will place it in the water bath. Okay, the tubes will sit in the water bath for 10 minutes. Again, I'm the only one that goes near the water bath, please. It's 100 degrees Celsius, I don't want you anywhere near it. Alright, so after 10 minutes is up, I'll remove the racks from the water bath. Okay, so once the tube is cool, you can get your tube, making sure it's yours, take off your cap lock and give the bottom of the tube a few flicks. Okay, then we're going to make sure our cap is secure, and you're going to place your tube in the microcentrifuge and give it a quick pulse to get all those sparkly Chelex beads down to the bottom. Don't forget the rules for microcentrifuge use. Okay, you're going to obtain a new tube. This is a new empty tube, and you're going to write your pin on it, lid and sides. Okay? When you remove your tube with the Chelex from the microcentrifuge, hold it up to the light so you can see the pellet of Chelex. The pellet is now bad and should be avoided at all costs. You are now in a parallel universe with the supernatant at its center. So you will now transfer 60 microliters of supernatant containing your purified DNA into the new tube with your pin. Holding the Chelex tube up to the light while drawing up the supernatant will make it easier to keep the tip of the micropipette in the supernatant layer and avoid the Chelex beads. So hold the new tube up to the light and make sure you didn't transfer any beads. Place the new tube in the designated rack and store it in the refrigerator until tomorrow's PCR adventure. Okay, so again, make sure we have no beads. We want to avoid those at all costs. And once you've got your 60 microliters of purified DNA into the new tube, you can discard all other tubes, cups, pipettes, wipe down your workspace with some disinfectant, and pat yourself on the back for a job well done. And we'll be ready for day 2. Hope this is helpful.